Okay.
Is it done now? Do you see?
Yes, we see you. We see your presentation.
Okay. How do you see it with all this stuff?
Yes, you need to put it into presentation mode.
Okay.
There we go. Very good.
Is it okay? Okay, I don't see anyone now, but that doesn't matter. So, okay. My name is Gernot Zissel again. I'm biologist and work at the Department of Pneumology. Yeah, what you see here is the building where our ward is located. Yes, the lab is a bit far away. You see it on the right side. So it's the lab is located in an industrial area because our former lab is deconstructed because all the building has to be rebuilt, and yeah, so we had to move.
Shoot, sorry.
Our department is part of the Medical Center, University of Freiberg. It's a rather small department. We only have 19 beds and a large outpatient clinic. So we take care for every kind of pulmonology patients, asthma, COPD, ILD, cystic fibrosis, hypertension, whatever. So all these stuff. We also engaged in lung transplantation together with the Department of Thoracic Surgery. So what we have and what our special interest is a large cohort of patients with interstitial lung diseases. So the main focus of our work was and is sarcoidosis, but also now lung fibrosis, and a bit broaden it to the ILDs. But working with sarcoidosis, you came across the fact that it is really closely related with berylliosis. So you have granulomatous disease of unknown origin. Okay, in berylliosis, the origin is clear, as you have the beryllium as a T-cell antigen. You have T-cell activation in sarcoidosis as well as in berylliosis. But anyway, from a clinical point of view, both diseases are not distinguishable. You have, regarding lung function, you have dyspnea, you have restriction, reduced DLCO in both sarcoidosis and berylliosis. If you look at the chest X-ray, you have vehicular lymphadenopathy. Again, in both diseases, you have increased lymphocytes, increased CD4-CD8 ratio. The granuloma in pathology, the granuloma look very similar. You have epithelioid cell granuloma. They are non-caseating. Again, you can't differentiate both diseases. So the only mean to differentiate berylliosis and sarcoidosis is the different T-cell activation antigen. In sarcoidosis, it is not known, although there is some work done that it possibly are autoantigens or microbes. That's not clear yet. But in berylliosis, it is clear it is beryllium. So this is just to demonstrate. You have at the left side a chest X-ray. You see here the increase in condensates. You have the same in CBD as in sarcoidosis. Yeah. So again, Beryllium Lymphocyte Proliferation Test is the only mean to distinguish sarcoidosis and berylliosis. The prevalence of sarcoidosis is very variable in Europe. So from north to south, it is decreasing. So in Sweden, it's rather 60 per 100,000. In Germany, we do have a prevalence of approximately 40 per 100,000. That means we have in Germany about 30,000 patients of sarcoidosis. with an incidence of 8,000 per year, approximately. There is no real data on CBD in Germany. This is just an estimation. Maybe we may have about 2,000 CBD patients, but there is no register, there's no really epidemiology done, so it is really unclear. Because we use this test to distinguish sarcoidosis and berylliosis, this has an implication. That means all tests, or nearly all tests we do, are performed in patients. Rarely we do have asymptomatic individuals where we perform the test. So if you have a patient and you have the question, is it sarcoidosis or berylliosis, you have to deal with that the immune system is already activated. You may have proliferative T cells, or you may have diminished proliferation, as in sarcoidosis, anergy is also reported. You may have therapy, and that is one of the problems we have, because patients use drugs to diminish to calm down the immune system. Steroids, for example. Currently, we don't know whether this interferes with the test, but we ask our patients to skip the therapy, at least to a very, very low dose. And we ask them to skip the therapy for about 4 to 6 weeks, but this has been done under the surveillance of a physician, not that the patients just don't take their drugs and the disease goes worse. And another problem is that you have to convince the regulatory bodies that it is necessary to do the test, and also the physicians to make them aware that it might be sarcoidosis, but it also might be berylliosis. The main argument is we don't have beryllium industry in Germany. So there is no industry, so we shouldn't have berylliosis. No industry means we don't have any mining, we don't have any mills or factories producing beryllium. But anyway, this is nonsense, because we have beryllium, because it is widespread in different kinds of industries. You have it in aircraft industry, atomic power, electronic industry, and so on. So beryllium is very widespread in any materials. And thus, all workers dealing with these materials may have the risk to get sensitized, and thus the sarcoidosis may be chronic beryllium disease. Some years ago, this fact was coming aware. So, and some guys tried to generate guidelines how to diagnose beryllium disease. And this is the title of these guidelines: Health Surveillance for Workers Exposed to Beryllium and Diagnostic Procedures in Beryllium-Associated Diseases. And this guideline was a milestone, because since this guideline has been published, we had the impression that the fact that we may have chronic beryllium disease in Germany becomes aware. It was present to the physicians, and they asked for the BeLPT. The guideline gives some hints. How should we do a diagnosis of berylliosis? So the main thing is the occupational anamnesis. Is there a known beryllium contact? If yes, we should do the BeLPT. Is there not directly known beryllium contact, but he's a metal worker, has dealing with metals in any way, and then it might be contain beryllium, so we should also do the beryllium test. And how this should be done, these steps in the diagnosis. So we start with an initial BeLPT, and if this BeLPT is not normal, we do a second split sample test, preferably in 2 labs. If one of these tests, again, is not normal or at least borderline, the chronic beryllium disease is confirmed. If not, then it is not confirmed. If one test, or the first test, is borderline, we do the same procedure. Again, split sample test, 1 or more is not normal or borderline, then it is confirmed. And if it is not normal, that means if it is normal, it is not confirmed. When the first test is normal and has no hint for beryllium sensitization, the diagnosis of CBD is not accepted. In fact, we do not use 2 different labs. All the split sample tests are done in our lab because in Germany there are only 2 labs working or using the BeLPT as it is requested by this guideline. At least we don't know whether there are other labs in Germany. Okay, it is more or less a technical talk. So I would like to present you how we do the test here. So normally you may know the use of H3-thymidine, which is a beta emittent. So you have to add the H3-thymidine to the proliferating cells. They include the thymidine, and when you wash away, you may have you use a scintillation counter, and you have the counts and can calculate whether you have proliferation or not. We do a slightly different test. We don't use radioactivity. We use a thymidine analogon, which is called brom deoxyuridine. This is also integrated in the DNA when it is proliferating, when the cell is proliferating, but it can be detected by a very simple ELISA. So we have an anti-BRDU antibody, which is commercially available, and you can detect the DNA, the BRDU integrated in the DNA just by ELISA. So the big advantage is that you don't need any radioactivity. You have no radioactive waste. You don't need expensive beta counters. Instead, you use a simple ELISA reader. The disadvantage is that you have a limited range because ELISA has a lower and upper limit. But to be honest, I never reached in the way we do the test, I never reached the upper limit of the ELISA reader. Yeah, it is, the test starts as very conventional. We take the blood, we isolate the mononuclear cells by a fecal gradient, wash and count the cells, adjust at 0.5 millions per milliliter, and use these cells in our proliferation assay. However, I much more like the second type of blood drawing. These are tubes we buy from Becton Dickinson. They have natrium heparin here. We have a gel plug, and the fecal is placed down here. You take the blood using this adapter and the needle, and then the tube looks like this. So this gel plug separates your blood from the fecal until you place it into the centrifuge. In the centrifuge, under pressure, these gel plugs get fluid, and then you have a normal fecal gradient. And after the centrifugation, it looks like this. You have here the plasma, you have here the PBMCs, again the gel plug, and all the red blood cells, granulocytes, they are stuck beyond the gel plug. And then it's very easy to harvest the cells. You don't need to separate an interlayer or something like that. And the second advantage is that at this step, the tubes can be transported up to 72 hours. And that's very interesting for us. You will see later. So we also use a medium with low protein. We don't use any FCS or something like that. We use a serum replacement solution to avoid unwanted T-cell activation by foreign proteins. And we use only 50,000 cells per well in a 69 well plate. So we need at least BE-specific T-cell ratio of at least 1 in 50,000. Yeah, we start with a 69 well plate. We add our cells, we add a positive control, and we add our beryllium solutions in a wide dilution step. So we're starting with 10 millimole and go down up to 100 picomolar. 1/3 of the plate is used only to detect the background. As I told you at the beginning, we do have patients. That means they might have background proliferation, and that is why we try to estimate what is the background proliferation of this patient. So the cells after 3 or 7 days, the cells are labeled with BRDU. And after 16 hours, we wash away the non-bound BRDU, harvest the cells, fix them, and dry for the ELISA. The ELISA is very simple. You wash the plate again. You split the double strand of DNA to make the BRDU accessible for the antibody. We use HCl at low concentration. For half an hour, wash the cells, add the BRDU peroxidase antibody, wash away the antibody. After 2 hours, add the TMB color reaction and measure. So that's it. What we get is such a matrix where we have our optic density measures. We have here the positive controls, the test, and this is our sorry, this is in the background. This is put into a calculation. Okay. We see here some outliers which have to be removed. And then you add these values in a spreadsheet for calculation. And you see here, we calculate the background proliferation by subtracting our background without BRDU and the background with BRDU. So we get here the difference, get a mean, calculate a mean, and standard deviation. What we have here is the mean plus 3 standard deviation. And this is the stimulation index calculated from this mean. That means this is our lower limit. This we calculate here, or we assume here that we need more than 3 in the simulation index to say this is not normal. You also calculate a second cutoff with the mean plus 2 SD, which would generate us the borderline. So every simulation in between 2.3 and 3 we would regard as borderline, and stimulation index above 3 would be positive. And in this case, we see here, this is a strong positive patient. In 3 of these concentrations, we have high stimulation index, which is far above 0.3, 3.0, and only in the lower calculations, or in the lower concentrations, we have lower stimulation index. If somebody is interested in a more comprehensive explanation how we calculate, we can do it afterward. Yeah. And that is what our patients or the physicians receive from the lab. It's just a sheet together with the with bar chart and with the numbers. So why do we do these detailed and sophisticated analysis? The first reason is individualization. Again, we have patients with an activated immune system, we have patients which are under therapy. So we feel that a general assumption that 2.5 is the limit would give us the wrong estimation. And that's why we calculate an individual background, an individual cutoff for the proliferation test. The second idea, or the second interest of us, is standardization. So I told you that in Germany, at least 2 labs perform this LPT as the guideline requested. This is the research center in Borstel in northern Germany, close to Hamburg, and we here in Freiberg. And to get a more standardized test and to ensure that we all use the same test, we try to generate a SOP, a standard operation procedure. It's not finished, it's not finalized yet, but we hope that this summer we get it finalized, and we will publish it. But first, we publish it in German, as we have to
So Dr. Zissel? Your audio is going off.
Regulatory bodies, but as soon as it is finalized, we try to translate and to make it open for, yeah. Hi.
Yeah. We're having a little bit of instability with the video connection. So.
Okay. Okay. So the background why we perform this SOP is that to ensure that both partners perform the same test, but also to enable other labs to perform this test and to justify the test for regulatory bodies, because they always ask what test you do. Is it really reliable what you're doing? So please show me your protocol and so on. Yeah, as soon as it is finalized, we also will translate it in English so that it can be published also internationally. Yeah, this is, I told you that I'm interested that the blood can be transported. We have very bad experiences when we get the blood just by the normal heparin or EDTA syringes. So these cells don't perform very well for LPT, but using these BD tubes after centrifugation, these cells are very stable and can really kept alive for more than 2 days. And this is important as we have some, very, how to say, labs or physicians which send us blood from Austria. There are two hospitals sending us samples. It's also from southern Switzerland. And they have to be transported at least overnight. And that is which mainly works in our setting. Yeah, and this is here in the north. This is the Research Center Borstel, which is located here in between Lübeck and Hamburg, which are also performing this test. Just to show what are the patients we have. So most of our patients have to do with metal working in a very general sense. Grinding metals, drilling metals, whatever, everything which makes dust from metals. We do have also patients working in a foundry or welding company. We do have a few dental workers, which are in Germany at least the original patients which are accepted as occupational disease that have been the dental workers. We also have a few patients working on electric devices, or very interesting, who had indirect contact with beryllium. That means spouses washing the clothes of their husbands or something like that. And far of all, metalworking is the most relevant occupational procedure to be at risk to have this chronic berylliosis. Only a few others have been positive and have really chronic beryllium disease. But as I said, nearly all tests are performed in patients. So we have done a few surveillance tests also. There was one aluminium plant in northern Italy. They were known to use material which contains at least some parts of beryllium. But during the workup, the aluminium was enriched, but also the beryllium was enriched. So we found several positive workers from this aluminium plant. And just a few months before, we published a cluster of beryllium sensitized patients or individuals who not really work with beryllium or metals, but we think is that they have been sensitized by environmental beryllium. So beryllium is the far most antigen we use in the LPT, but we use this test also for other metals, sometimes aluminium or zirconium. Very few we use it for cobalt and chrome, but we also use it for drug. It is known that there are some drugs or some diseases are based on drug sensitization. Mainly it's amiodarone. but also methotrexate, nitrofurantoin. I moved 10 years ago. I moved from north to south, and in northern Germany, the hospital was a referential center for mycobacteriosis, and we had a lot of patients reacting against the anti-tuberculosis drugs like Erenfat, rifampicin, isoniazid, and so on. And we used also the BeLPT to get to evaluate, are these drugs are the reasons for the lung disease in addition to tuberculosis. This has also been published, in the last century. But here in southern Germany, these anti-tuberculosis drugs are not of relevance anymore. Yeah, okay, thank you. That was what I have to present, and I'm open for questions.
Well, thank you, Dr. Zissel. That was great. I think we can stop sharing your screen. Can everybody hear me? Yeah.
Yes.
Okay.
Yeah, I want only to add something because we are doing the same test with the protocol of the National Jewish. So if someone asks, what is the test that you are doing? So we have the backing of the guidelines, of the ATS guidelines, as we are doing exactly the same protocol. So this gives us a backing because I think that you are the only one not using thymidine. So I understand that you must have a very accurate SOP, as you are the only one doing with not doing with the thymidine.
Not the only one. We had a discussion on publication of the in chest. So some guys also said, you don't use thymidine, you use this BRDU, so it's not really accepted, and it's unusual. And they sent us a few literatures, and 1/2 of them use also BRDU and not thymidine. So we're not the only.
We're doing also the MELISA test that is a thymidine based, so it's easy for us, or MELISA or BeLPT. All the two tests are done by thymidine, so it's easy for us to go from one test to the other and not installing some other kind of test. But I agree with you. There's no radioactivity, and that is the main point. But we have all a lab step with all the methods that we can avoid any contamination or radioactive exposure to the workers. And the thymidine that we use is very, 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 very low concentration. So I think that the thymidine is more sensitive, as you told, than the ELISA test.
I really don't believe that it is more sensitive, but you have you have a certain range where it works. But in our hands, we never reached the upper limit of this range. So depends on the technique.
Another question that I have: you told that you are doing 3 days or 7 days. You are not. You are doing the balls.
Yeah.
We are not. Yes, the 7 days for us give a lot of rush. The background. So we are doing these 6 days, 4 and 6 days. 7 days give us a lot of background, rush background. So I think that the 6 days is working for us better.
Okay.
Okay. It's good that you have in Germany another lab. We don't have in Israel another lab doing BeLPT, so we cannot split our samples.
In fact, we don't split. We don't send the samples from southern Germany to the northern part of Germany. So we do it in one lab. So it was requested by the group generating this guideline. But in fact, we need more labs doing the LPT also in Germany.
All over the world, not only in Germany.
Right.
It's a problem.
So I have a comment from Dr. Cullinan, who had to leave early. He said he was a lovely talk, many thanks, and he found your recent chest paper journal article fascinating. I got a comment during the presentation by, I'm assuming Lisa Barker, that they find extra pulmonary granulomas in sarcoidosis patients, but those are rare in Beryllium disease.
Yes.
Okay.
That's indeed. We had a few patients with Beryllium disease with, good question, what was it? Eye involvement? I don't remember. It's rare, but there are extrapulmonary manifestations of Beryllium disease, but very rare, that's true. But anyway, in sarcoidosis, extrapulmonary disease is also not that common. You may have, but it's more often you have the lung involved.
Right. So does anybody have any comment about the, go ahead.
I have two remarks. Your paper in chest of the disease being environmental, it's a great paper.
Thank you.
Yes, it's a great paper because it's the first paper showing that the disease can be environmental and not occupational. I enjoyed very much this paper. Another, there is a paper was published just in June in chest that is called Sarcoidosis is an Occupational Disease. That is a great paper. So that perhaps there is no disease that it's called sarcoidosis. That's that we not know the antigen. We cannot call this idiopathic. That is a great paper. Perhaps I will bring this paper in my talk in September, because it's very the first time that there's a review calling the sarcoidosis not an idiopathic disease. And in this paper, they speak a lot about beryllium, BeLPT, and also about the MELISA test. So perhaps I will bring this paper in my talk on September, because it's very important.
That would be great.
There is no sarcoidosis, perhaps.
Yeah. So if we don't know that beryllium is an antigen in beryllium disease, it would be called sarcoidosis, because you have no means to differentiate. And the same, I'm convinced that sarcoidosis will divide more and more. So maybe it's autoantigens, maybe it's really what the colleague from Japan favors, it is Propionibacterium, possibly. So we have to wait and to search for antigens in sarcoidosis.
Yes, it's not right to call this an idiopathic disease. That is the point.
Yeah, idiopathic, no, because you have T-cell activation. That means you must have an antigen. So
you must have the antigen. So, and that we don't know the antigen.
Exactly.
It's different. So, idiopathic is that there's no reason.
Concerning occupational disease, there is, I think 2 or 3 years ago, there was a paper published by colleagues from Switzerland, and they analyzed the epidemiology of sarcoidosis of all these parts of Switzerland called canton. And they have seen that it's not equally distributed. So if it is really idiopathic, I would expect that sarcoidosis is spread equally. But they found spots with high incidence and spots with low incidence of sarcoidosis. So that makes me, and they also analyzed what is the main product of this area. And that was interesting to me, because it gives some hint that indeed, yeah, it is related to occupation, to pollution, whatever. I agree. So that would be an interesting part.
So can you tell me what paper that is, and maybe we should send that out?
The paper is in chest, June, the 2 of June 2021. That is from Ontario. Christine Oliver is the first author.
Okay, so Dr. Zissel, you still have your screen sharing.
Sarcoidosis, an occupational disease. If it's an occupational.
Okay, I'm sorry, Lizzie. I was a little distracted. So I know the one that Dr. Zissel mentioned. What was the one that was the sarcoidosis patients and their geographic locations?
The first author is Deubel-Weiss. I can send you the paper.
Okay, if you would do that, I would appreciate it. I'll send it to the group as well. Okay, well, I thought that the tubes that you're using, and they seem to work for you, are kind of a breakthrough, especially in the European areas where, in the US, it's real easy to get a sample sent overnight. But this is cause for great concern, I think, for us in France. And I think maybe in other countries as well, is that we don't have that reliable of a service for overnight shipment here. And so I think that's a great improvement. I don't know what other people would think about the test too.
Yeah. Exactly. We use them for the ELISA as well, but as we depend on higher volumes of blood, we had to use 6 of these tubes for one patient. And this is really a cost factor because, as far as I remember, one of these tubes is about 10 euros.
A bit less, but anyway, yeah.
Yeah. And so I don't know how much volume they take. They take some
8 milliliters.
8? Okay, great. And we try to use the longer ones. They are packed in 2 different sizes, but they don't fit into the centrifuge.
Exactly.
They fit, but you shouldn't use the centrifuge because then they break. Yeah, I had really good experience with those tubes, but it was just a matter of costs.
Yeah. So because we all have patients, so they are requested to do the test because if it is sarcoidosis, it has to be paid by the health insurance. If it is an occupational disease, we have another body who pays for. So the regulatory bodies have their own interest to perform this test because maybe they get rid of this patient. So they pay. They pay the test, they pay the amount for the tubes because they may avoid the cost for the patient.
Okay. Yeah.
That's the main reason.
And Catherine is absolutely right, especially in times of COVID and Corona. We have severe trouble with shipments from US, from different countries, even from UK to Germany. They always stuck in the customs, and so it's really hard to get them in time from Germany. It works with some express service, but overseas it's catastrophic.
Yeah, we have the same experience now for Switzerland because they cut the negotiations with the EU, and from this we had really problems to get the samples into Germany.
Yeah,
they stuck, as I said, they stuck at the customs for weeks. Until I said, please throw it into the trash. I don't use the sample anymore. Yeah, it's really hard. Yeah.
Yeah.
No problem from Austria, but from Switzerland, I think it will not work anymore.
Interesting. Well, I think that was a great talk, Dr. Zissel. I really appreciate you presenting your method. That's really great that we're all sharing on this. So I don't have